great pleasure to welcome you to our spring 2021 meeting. As you can see on this slide, that's a little bit of a teaser. That is the cover of this year's annual water quality report. And Sheila Gordon-Delane and the team have worked really hard. It's another great report. And this year not only includes what's happened in the last 12 months, but it also is a look back over the last 20 years, because this is our 20th, last year would have been our 20th anniversary, and this is our 20th book. So sincere thanks to Sheila and the editorial team for doing their usual amazing job in pulling this all together. I'd also like to take this opportunity to thank Anna Courier, who helped to work with the group to pull together today's presentation and is this morning's slide master. So thank you very much, Anna. And I would like to also thank Kimberly Ong, who is this morning's Zoom master. And Kim, I can't tell you how pleased I am that you're doing that and not me. So thank you very much. Next slide, please. Here's this morning's agenda. We're going to cover a little bit of housekeeping, and then I'm going to turn it over to Ed, and he's going to share his opening remarks. Then we're going to do some sort of quick snapshots of the projects that we're working on. There are 5 different projects that will give you a sort of a one-slide update, and there will be an opportunity for Q&A. Then there will be the dockside chat with Dr. Bob Bailey. We're very pleased that the Bob has sort of done the inaugural for us on that this year. And then we will have Q&A on Bob's section if you've got some stuff you'd like to ask him about his research, and then we'll close. Next slide, please, Anna. In terms of housekeeping, if you could please, if you've got questions, we'd really like to make this as interactive as we can today. So please, if you've got questions, input them, your questions, your comments into the chat line, and we'll sort of keep track of that. Please mute your mic when you're not speaking in order to sort of minimize any sort of background noise. Please shut off your video except when you're speaking. The reason we're going to ask that, because every video takes some bandwidth, and some of the people who are dialing in this morning are dialing in from their cottages where the black flies are. And unfortunately, some of the, as you know, the bandwidth out in the country can be a little bit constrained sometimes. And if there's not as much video feed, then it doesn't consume as much bandwidth, and they'll have better luck with their signals. So if you're speaking, then by all means come back in with your video and your mic, but otherwise, if you could keep them off. And finally, this morning's meeting will be recorded so that we can put it on our website for those folks who would have liked to have joined us but weren't able to. So next slide, please, Anna. I'd like to take this opportunity to acknowledge that the Greater Peterborough region is in the traditional territories of the Mississauga-Nishnabeg Territory, and in the context of the Rice Lake Treaty 20 and Williams Treaties. We know everybody who's on the call here today is very concerned about water, and that there's much that we can learn from the First Nations peoples in terms of water protection and working towards a more sustainable and regenerative future. So thank you for all that you've done for us. Next slide, please, Anna. At this point, I'd like to turn it over to our KLSA chair, Ed Leardom. Ed, over to you.
Thank you, Tom, and welcome everyone to our annual spring meeting. It's great to see you all, and thank you for joining us today on this beautiful day when we do want to be outside feeding those black flies. I'd like to start by recognizing all of our volunteers and supporters. Today, you will hear about our many projects we are currently doing and planning, and we could not do any of this without you, our volunteers and supporters, so thank you. Last year, and I call it pandemic year 1, as we all know, was a decidedly different year for everyone. For KLSA as an organization, like many others, it was a year of not knowing if we could continue our annual activities and the programs we had planned to carry out in the summer of 2020. And we never had a virtual meeting before. What the heck is Zoom? The world changed quickly, and we adapted as best we could. At first, we used a conference calling system for our board to meet, but later we adopted Zoom for our meetings. But unfortunately not in time for our usual spring public meeting in early May, which we had to cancel for 2020. But now, a year later, We are now well versed in holding meetings online using technology and tools like Zoom as we are today. All our board meetings for well over a year have been virtual, as was our AGM last October, with only a few people in attendance. Not like the 100 or so we normally have at our public meetings, and not like the almost 50 we have this morning. So thank you again for joining us today. At that virtual AGM meeting in October, a new board was elected. A few members of the 2019-2020 board wished to move on to other endeavors. Kathleen McKenzie, who was one of the founding members of KLSA and vice chair for many of those years, and managed the E. coli testing and phosphorus testing programs. Lynn Woodcroft, on the board since 2013 and our secretary since 2014, and Doug Colmer, on the board since 2016 and contributing to many of our programs. Since then, Tracy Logan, on the board since 2014, and Bill Napier, also on the board since 2014, and serving as chair for until this past October, have also gone on to other endeavors. We thank each and every one of these good people who have contributed to our efforts to help ensure we have good, clean water in our lakes and streams. Today's board, KLSA board, is made up of me, Ed Leardom, your chair and treasurer, serving a dual role, Tom McAllister, our vice chair, Carol Cole, our secretary, Sheila Gordon Delane, our recording secretary. Remaining directors are Bob Bailey, Anna Courier, Jeff Chalmers, Mike Dolby, Cindy Lee, Kimberly Yong, and Brett Traguno. 2020 happened to be KLSA's 20th anniversary. It all started in October 2000, right after, sorry, I think, did I say October? August 2000, right after, as a result of the Walkerton, Ontario E. coli outbreak from contaminated drinking water in that community. We were disappointed we could not celebrate this milestone with you. Our annual 2020 lake water quality report, about to be published in this coming week, celebrates our 20th anniversary with special articles, memories, and pictures from our archives. Wave 1 of the virus seemed to wane as summer 2020 started, and our volunteers could do our E. coli testing with special protocols at the lab in Lakefield. However, the Ministry of the Environment and Conservation and Parks Dorset Lab remained closed until the end of the summer. So the LPP program was suspended until the time when the last sample could be taken in October. This year, the latest information we have is that the program will start as usual and continue for 2021. We piloted our 3-year continuous water temperature and dissolved oxygen monitoring program, set to fully launch this summer. Why do I have October in my mind? We initiated a Starry Stonewort and invasive speak aquatic plant location identification program, which will continue this summer. And in partnership with Watersheds Canada, we initiated their Natural Edge program in the Kawarthas, assessing 5 shoreline properties on Lovesick Lake and Big Cedar Lake. And this program continues this year as well. You'll hear more about all of these initiatives in this meeting with a chance to ask questions about them and even volunteer to participate as a volunteer community scientist. In 2020, our biggest challenge was how to distribute our annual lake water quality report. We had all the articles and data prepared, but once printed, not having a spring meeting in one person and restrictions around meeting people and moving around, we had to figure out how to get the reports to you. We did delay the printing of it until later in May, but did publish it up on our website as soon as we could. Printed copies were offered as mailings to our members, but the bulk of the copies were placed in businesses and other locations around the Kawarthas, which were open to the public. We still have a few copies left. If you wish to have a printed copy of last year's report, please contact us by sending an email to our new email address found at the end of this presentation. You can always view and download this and any other report from our website. We have the same challenge with this year's report. So you will be able to find this year's report on our website in the coming week, and it is going to print very soon. If you want a printed copy, please contact us again at our new email address found later in the presentation. We will send an email out to our distribution list when it's available, and you can reply to that email as well. We will also be dropping them off at some of the local businesses in the Kawarthas, which are open to the public. The last part of our meeting today features Dr. Robert Bailey, professor in the Faculty of Science at Ontario Tech University and one of our newest KLSA board members, sitting on his dock at his cottage in Lower Buckhorn Lake and talking about how he studies aquatic plants in our lakes. We hope you enjoy it. I'd like to thank you for your continued support, both financially and as volunteers. Without your continued support, we could not be able to keep a close eye on the quality of the waters in the Kawarthas. My thanks to the KLSA board members who put this meeting and Dockside Chat together. Please enjoy the rest of the meeting, and we welcome any comments which you can send to our new email address found at the end of this presentation. Thank you very much. Back to you, Tom.
Great. Thank you very much, Ed. I really appreciate the update and for all that you do. As wearing one hat is bad enough, wearing two as both the chair and the treasurer is a big job. So thank you very much. Next slide, please, Anna. We're going to shift now into project snapshots, and we'll just spend a few minutes on each project. It doesn't do them justice, but just sort of reminds us all of what we've got on our plate. And as Ed often likes to say, we collectively, all of the volunteers, including the board, are a busy bunch. Next slide, please. Now, as I said at the outset, we would welcome your questions here this morning, and so please do do go ahead and put them into the chat, and there will be a window right after we go through sort of the 5 snapshots, and you'll be able to get your question asked and answered. As Ed mentioned, Kathleen has moved on from the leadership of the E. coli team. The new team this year is going to be Cindy Lee, who joined us relatively recently, and Mike Dolby, who many of you will have known for many years. The protocols this year, I'm pleased to say, are going to be pretty consistent with in the past. As Ed pointed out earlier, sort of 2020 was a year where we didn't know what we could do, when we could do it, or whether we'd be able to even access the lakes and so on and so forth. So long story short, it's kind of more like business as usual this year. Initial communication has already gone out at this point in time. And as Ed mentioned, we would welcome additional samplers in the western part of the Kawartha Lakes region. We've got great coverage in the central and eastern parts, and we'd like to augment the coverage that we've got in the western Kawartha Lakes. So if you're interested, we'd love to bring you on board. So please reach out to, as you can see at the bottom there, Cindy, and there's her email address. The chain of custody, the details on that has been sent out in our emails. It's also posted on our website. The cost this year is unchanged, but the way that you can sort of straighten up for the testing on your lake is changed because we now accept e-transfers. So that'll make it a little bit more convenient for everybody. All of the information you need is available. We'll point out that the bottles have to be picked up at STS in Lakefield, and you'll have to phone in advance for an appointment just because of COVID-19 protocols. So very much looking forward to another robust year of E. coli testing and under Cindy and Mike's leadership. So thank you very much to Cindy and Mike, and thank you so much to all the volunteers who have been doing this program for so many years. Next slide, please. Starry stonewort. This is something that chances are a few years ago you wouldn't have heard this at all, but it's becoming more prevalent, unfortunately, in our region. And we're working with Kawartha Conservation as well as with Dr. Andrea Kirkwood of Ontario Tech, as well as the Starry Stonewort Collaborative. And our objective in this project is to get a better understanding of where it is and what the relative abundance is of this particular invasive species. Community scientists, community volunteers will do the sampling bi-weekly from July to October, and then they will identify the macrophytes in each sample. As a non-biologist, I would just call them the weeds, but I'm advised by people who know these things that they're not really weeds. Many of them are just aquatic plants and macrophytes. But the good thing is that now with the benefits of the training that you'll get, you will now not just be able to curse your weeds in general, you'll be able to single them out and identify which ones you dislike the most. Starry stonewort has been identified as an important invasive species. We will report the findings of starry stonewort into a database that will enable the group to get a sort of a picture of where it is and where its relative abundance from the July to October period. In terms of training, it will be provided by a webinar in June, and it will show you how to go about it, how to identify it, and so forth. KLSA, Kawartha Conservation, and the Starry Stonewort Collaborative will provide ongoing support to all of the volunteers. And so, once again, if you'd like to assist with this, we'd really appreciate it. Next slide, please. And if you'd like to participate, please feel free to reach out to Carol or to Brett at these contact points. That information will be available elsewhere in the presentation as well. So thank you very much to the volunteers who are making that happen. Next slide, please. Okay, phosphorus testing. As Ed pointed out, this was a program that was postponed or canceled for the first part of 2020, because once again, we sort of didn't know what we could do and when we'd be able to do it and whether labs would be open and so forth. Kathleen has led this initiative for many years, along with the E. coli program, and she's rolled off this. Similar to E. coli, this project will be led this year by Cindy and Mike. And in terms of how it will run run pretty much as it has in previous years, pre-COVID. So we would appreciate if you were a tester for phosphorus, if you could please reach out to us and sort of connect if you have not already done so. And we once again would welcome additional volunteers who could assist us with this program this year. Some of you will recall, some of you did some testing in late 2020 when the lab at Dorset reopened. That information has been gathered and the results are being analyzed, but I'm pleased to say that there is an article rather in this year's water quality report giving a bit of an update in terms of where that stands. So the primary contact of that once again will be Cindy Lee, and her email address is right there. Next slide, please. In terms of the water temperature and dissolved oxygen project, as Ed mentioned previously, this was a pilot project in 2020 under rather interesting circumstances, but we're sort of getting up and running this year and really finding our rhythm on this. It is intended to provide a baseline for climate change tracking and modeling, and we can see climate change. We can feel it in terms of ice in, ice out, and all of those things. And the water is a great sort of indicator for how much additional energy is being absorbed in our region. This will be a 3-year study. It will be conducted via data loggers, which are these just small data, almost the size of like a toonie, that will be attached to buoys in the Trent Severn waterways and on nearshore places like on the legs of docks and so forth. And we will also be sampling the deep basins. The Kawartha Lakes are pretty shallow bodies of water for the most part, but there are some deep basins, and they will provide us with additional insights into the water column and the characteristics of it, which of course are critical to aquatic life, whether it be animal or vegetable. So very much looking forward to the rollout of this program this year, and to your support of this particular initiative. And thank you in advance to the volunteers who assisted with it last year, and to all the volunteers who are rolling it out this year. Next slide, please. Okay, the Natural Edge program. Bill Napier had been leading the charge on this, and he's rolled off the board. But we want to take this opportunity to thank Bill for his leadership on this initiative and on so many initiatives in Kawartha Lakes, and in his role as chair over the last sort of, I guess, got to be 6 or 7 years. So we greatly appreciate Bill's leadership on this and so many other initiatives. The new Natural Edge program coordinator is Kimberly Ong. And she just joined us on the board in this year. Now, the purpose of this project is to perform 10 shoreline restorations, and each of them will have about 100 plants put into place through the Watersheds Canada Natural Edge program. And Watersheds Canada is a partner with KLSA, and I want to thank them and all of the organizations that we partner with, because that's how we get a lot more reach and a lot more impact. So thank you to them for their support. In terms of how the program is going to unfold this year, between sort of May and June, we'll plant the first 5 sites. Now, these sites were assessed last year, and the planting plan was developed for each of the respective sites last year. In June of this year, we'll identify property owners for the additional 5 sites that we would like to do. The way this works is that it costs $500. Like the overall cost is about $500 per restoration site. 250 of that, $250 come from Watersheds Canada, and the other 250 comes from the land or the property owner themselves. So that's how the funding works. So in June, we'll identify them. In July, Watersheds Canada will assess those sites and develop the planting plans for those respective sites. And then in August or October, we'll work on those sites as well. If you're interested in participating in this project, either as a property owner who would like to explore having this program implemented on your property, or as a volunteer, please reach out to Kim Ong at the contact information you see at the bottom of this screen. Next slide, please. Now, those are sort of the 5 snapshots, and we would like to give you an opportunity to participate a little bit more directly. If you've got any questions on any of those 5 programs or any other programs for that matter, now would be a great time. And if you'd like to address your question directly, in terms of if you'd like to, what you can do is what I'm going to do now is ask Kim if you could kind of sort of capture what questions we've got there. You've got them on chat, and if the individual who's got the question would like to actually ask the question themselves, verbally, then what they could do is just unmute to themselves, and they could put themselves on video if they so desire as well. And we'd like to have a bit of a conversation. So Kim, let's see what the questions are this morning. Over to you.
Thanks, Tom. So we do have some discussion going on in the chat. Looks like we have an offer for some volunteers for the water testing from Martha, who is the president of the Kawartha Park Cottagers Association. So thank you, Martha. And she was asking about asking maybe to get some clarification regarding the logistics and separation of the projects, regarding who does the water testing for phosphorus versus E. coli. And I see Jeff and Cindy have been answering a bit here, but I wonder, Martha, if you wanted to unmute yourself and kind of ask any follow-up questions or reiterate for the whole group.
Hi, everyone. Thank you so much. So yeah, we have a new water tester this year. Judy Finch has been doing it for us for a little while. And so the whole thing is kind of new to me. And we want to get our new water tester set up. So I'm thinking that she has been doing the E. coli sampling. And I don't think KPCA has been doing any phosphorus testing. I'm wondering, do we want to do the phosphorus testing? Is it an extra charge to do the phosphorus testing? Can the same person do both and take the water samples into Lakefield at the same time? How might you set that up?
Cindy, would you be able to, Cindy or Mike, perhaps you could respond to that?
Yeah, Mike Dalby here. Well, I believe that we have quite a lot of phosphorus testing already on Clear Lake. Jeff Chalmers, I believe, does that. Is that right, Jeff?
That's
Anyway,
correct.
perhaps you.
And I think Stony Lake is covered fairly well too in terms of locations, Martha. So that's probably being done with the Association of Stony Lake Cottagers.
Okay.
Or
All right. So
So-
then I'm not going to worry about it.
No, Kawartha Park, of course, has been you have consistently done E. coli testing, and we appreciate that. So if you let us know who the new tester is,
Yes.
that would be very helpful so that we can ensure that what happens is your tester takes the samples to SGS in Lakefield, and SGS send the results to Cindy and myself, and we distribute the results to you. So as long as you let us know who the results have to be sent to, we'll make sure you get them.
Okay, so Mike, mike.dolby@klsa.info. I will send you that person's contact information.
That would be great.
Great, thanks,
And you can
everybody.
CC Cindy too. Thank
Okay,
you.
thank you.
Okay, Kim, next question.
Great. So we have a question from Carol Armstrong. And the question is, I think, something that's on lots of our minds. And Carol Cole, maybe this one's for you. Most importantly, what is being done to slow the spread of stony starwort?
Well, to slow the spread, the only thing we can do mostly is to avoid spreading it ourselves. The starry starwort is spread primarily by people and watercraft. So for me, the best thing to do is to figure out where it is, which at this time we don't always know, and then try to avoid those areas with our motorboats mostly. In the States, that's sort of what they've been doing. And right now there's not a way to eradicate starry starwort, so not spreading it is the most important thing. And to not spread it from lake to lake, the obvious and most important thing is using common sense and clean, drain, and dry that we've been promoting for a really long time. Take your boat out, make sure there's no weeds or macrophytes on your boat trailer or trap between them. Empty your fishing water in the back where it belongs, empty your bilge, dry your boat, let it be out of the water for about 5 days before you put it in a new lake. All of the lakes that are infested have basically, for the most part, been infested by watercraft just traveling from one infested lake to the other. So that would be the best thing to do, is just clean your boat and stay out of patches of starry starwort if you know that it's in your lake. And that's about it for me.
Great advice, Carol. Thank you.
Welcome.
I think the next question we have is from Brenda. Brenda, do you want to unmute yourself and ask a question?
Yes, the Natural Edge program, just wondering what is covered in the 250 from the participant and 250 matched amount, because, you know, if we have already identified people ourselves, for example, we work with an Indigenous plants person. I'm with Truth and Reconciliation Bob Cajun. We're working on a Bob Cajun Beach project with this person who is an expert in plants. So I'm just wondering if on our own properties we can hire the person ourselves, or it has to be somebody from that program.
I could take this question on, Brenda. Thank you for the great question. So I am new to the transfer of this project to me as project manager. So I'm not exactly sure whether or not you can identify your own expert. The program currently is set up to with Watersheds Canada, who has an expert on staff, Chloe Lejoie, who, as part of the program, comes out to do kind of a site visit with the landowner and kind of look at the site and actually develop a planting plan. And this person will do a survey, and then they'll actually produce a site-specific plan, a planting plan that they would provide you. And also, along with that, also provide a starter kit, which includes coconut mat, mulch, bare root plant, potted plants, and wildflowers. And then we would help you plant those. However, if you have your own expert, I will look into it and I will get back to you if you maybe leave me your email about whether or not you can identify your own expert, and we can work with that as well. That sounds great.
Okay. I'll put my email in the chat. Thank you.
Perfect. Thank you. Thanks for your interest.
Does anyone else have any questions?
I see Doug Colmer has their hand up.
Okay.
Correct. I would just like to be in contact with the person that had asked about if there was anything that could be done to slow the spread of starry stonewort. If they would take my email address and send me an email, I'll be in contact with them as to what we have endeavored to try and do to control it in Big Cedar Lake. Is the person still on the line? My email address is dougcolmer@gmail.com.
Okay, well, hopefully we can make a connection there. Thank you, Doug.
Okay.
Tom?
Yes, Kim.
I don't see anyone else with their hands up. And I will continue to monitor the chat if anyone else wants to pose questions.
It's Jeff here. I was just going to make a comment and ask Carol. I know that they did some signage in areas of starry stonewort in Stony Lake, warning people. Is that signage campaign continuing, identifying the other areas? And because boaters won't know what's below the water. And
Yep.
And so I was just wondering if you'd let the group know what's been happening in Stony Lake,
Sure.
when new areas are identified.
Okay, so in Stony Lake, we've actually, because there's a few of us that have been looking at the starry stonewort for a few years, so outside of KLSA. So we did do signage on our lake, and we shared that signage with Big Cedar Lake and also with Lake Scugog folks. So what we have done is mark areas, like we have the Lost Channel right by my cottage, actually, and we put signs at each end warning people to stay out of there. Down at Gilchrist Bay, there's signage down around there, and there's some signage in the duck pond. But what we found is that the spread in Stony Lake is so extensive now, especially down near Gilchrist Bay, because it's boated through often down there. There's so many spots now that making individual signs for each spot isn't going to work. So there's a group of people on Stony Lake that are actually talking about different ways of marking it. And one of the things that we were told by Doug Comer actually last year on Big Cedar Lake and Diane Truzzi's group is that they actually are marking it with little, like the Javik bottles that we would mark a rock. So so there's talk of doing things like that on our lake and kind of publicizing it more. On Stony Lake, the Environment Council also put up warning signs at every boat launch that there is starry stonewort in this lake and that it is spread, through watercraft primarily. So they've signed everywhere around here, and we're kind of hoping that people who boat in and out of Stony Lake are paying a little bit more attention when they leave the lake. I know on Big Cedar, they have done a few different things that we haven't tried on Stony Lake. So getting in touch with Doug would probably not be a bad idea. The other thing I didn't mention was, in the United States, a lot of the research coming out now is talking about hand harvesting, not mechanical harvesting. You don't want to take big mechanical harvesters into an area by your dock and taking out all of the other native macrophytes. That's one of the problems. People do go in and they clear their swimming areas, and that's a place that starry stonewort loves to go. So my advice would be, keep your little swimming areas small, watch around your dock to see, make sure you identify it early. And then if you do find starry stonewort and it's identified accurately, you can, gently hand harvest small patches. And that's something that's being more and more accepted in the United States. And I think it will be here too eventually. So, but again, we're on the Trans-Severn Waterway. You have to be careful when you do that. You have to be careful how you do it, and you'll need a permit. So.
Carol, I wonder if you could, you mentioned harvesting it gently. Could you please tell the audience, why does that matter? Like, what is the nature of it?
Okay.
Sure.
Sure.
What happens if it gets chewed up? Why does it matter?
If it gets chewed up, Doug's got his hand up, Doug knows. It spreads by fragmentation, and there's tiny little white bulbils that are on it later in the season, and they float off if you ripped it out and just let it float away. Every little complete cell will start an entirely new patch of starry stonewort somewhere else. And so boating through it constantly, you're cutting it up and you're spreading it. So that's why we want people to stay out of it. Fragmentation and the spread of the little bulbils is how it's going to spread within an already infested lake. And if on Stony Lake, the map that the Environment Council is going to publish shortly showing where it is in our lake, you can see the extent of the spread. And when we first found it, it was in two very sort of isolated areas, and we knew exactly where it was. But now, because of the boating and things, it has really spread. So, yep.
I see we're still okay for time. We've got time for one more question. Kim, I see there's a question there from Peter Raymond. Peter, would you like to unmute yourself? I think, Carol, this will be another one for you.
Sure. Oh, does Peter want to ask? Or his question that I can see on the screen is, are there programs to promote cleaning boats before going in and coming out of lakes at boat launches and across the Kawarthas? Well, the invasive species awareness program folks have been promoting, clean, drain, and dry, which is usually when you remove your boat. So there hasn't been anything that I've heard of promoting it before you put it in. But as long as you clean the boat, I don't know if that matters much. I would say clean it when you take it out, then it has a chance to really be dry. Starry stonewort, the little bubbles and fragments do not last that long out of the water. So if you're leaving your boat clean and dry for 4 or 5 days, you'll put it back in another lake without having to be afraid of transmitting starry stonewort. And it's not just starry stonewort, that's the thing. A lot of invasives are spread by boats, Eurasian water milfoil, all sorts of anything that spreads by fragmentation. So everybody around here is, very aware of Eurasian water milfoil and the problems that causes. Well, that's nothing compared to what starry stonewort can do in your lake. So, be conscientious, clean your boat. And that would include kayaks, canoes, whatever you've taken through a lake. Make sure you've cleaned it before you put it anywhere else.
Good. Great. Thank you very much.
Okay.
Go ahead.
Yeah, that's right.
Go. No,
that's okay.
Carol, go.
I was going to say the other thing. Right now, there's a change in the Invading Species Act that is being put through, and they're asking for comments. So if you want to look at that, you can look at the MNRF website, and there's actually a little place to comment on legislation that would make clean, drain, and dry actually a regulation that's required, which would be really great, because up until now there really isn't. It's a suggestion, but there's no law saying you should do it. So if the legislation gets passed, perhaps we'll start to take this a little more seriously and provide things like boat wash stations, which is my dream. To have boat wash stations at public boat launches would be great. So you have a chance to comment on that, I think, until June 7th. So.
Excellent. Thank you. Well, listen, I see actually Bob Bailey has just asked a question. Carol, could you put that website URL here in the chat room? So if you could do that.
If I can figure out how to do it, I will. And if I can't, I will get it posted on our Facebook page and our website so people can find it.
Good stuff. Okay. Listen, if I could suggest that we now move to the next part of the presentation. Thank you very much to everyone for your questions and engagement, and thank you to the members of the board for their responses. I'm going to turn it over now to Dr. Bob Bailey. Bob, over to you.
Thanks very much, Tom. And I think one of my colleagues, Anna or Kim, is going to play this little video that I made, and be happy to talk to folks about any questions they have afterwards. So take it away.
All right. Can everyone hear me?
Yes.
Okay. So just let me know if the audio doesn't work, but it should be all right.
I'll give you the thumbs up, Anna.
Can you hear it?
I can't hear it or see it yet.
You have to share your audio as well as your video. And when you're sharing your screen, there's a spot to share your audio on your computer.
Yep, just 1 second here. I'll do that right now. All right. Yeah, we checked this before. How's that?
It's good.
Great.
Well, welcome everybody, and thanks for being here for our first dockside chat. My name's Bob Bailey. I'd like to begin with a land acknowledgement. For those of you who haven't experienced this, I know most of us have. This is really important for me, especially with the kind of work that I do and what we're talking about today. I'd like to acknowledge that I'm standing on the traditional territory of the Anishinaabe First Nation, just a few clicks from Curve Lake First Nation, our neighbors here on beautiful Lower Buckhorn Lake. So thanks for the welcome that we've had here. The work I do, it's important to me that it really speaks to the communities of this area, particularly the Indigenous communities. Well, thanks very much to the Kawartha Lake Stewards Association for giving me this chance to tell you a little bit about the research I'm doing in the Trent Severn Lakes. Before I do that, I wanted to go back a few decades and just give you a little bit of a window into how I got started in looking at freshwater lakes and streams. About 40 years ago, I did a master's with Jerry Mackey at the University of Guelph. Jerry's a great freshwater malacologist. He works on clams and snails. And at the time, interestingly enough, I was working on fingernail clams, these little couple of millimeters long clams that live in ponds and lakes. And one of my field sites was at Birdsall's Beach, just across Rice Lake from where I live now in Harwood, Ontario. So I did the master's with Jerry. I did a PhD at Western in the 80s on the pre-zebra mussel mollusk community in Long Point Bay and Lake Erie. And then I stayed on at Western for over 20 years as a biology and environmental science prof. I worked on everything from those same zebra mussels in the Great Lakes to the effect of gold mining on streams in the Yukon to the effect of intensive agriculture on streams in southwestern Ontario. And a couple of years ago, I was lucky enough to get back to what I think of as the best job in the world, working as a prof, doing research on lakes and streams. Coincidentally, back in 2012, my family bought this wonderful place on the shore of Lower Buckhorn Lake, and I started getting interested in the Trent Severn as a system and Lower Buckhorn in particular. One thing I noticed was the weeds. Everybody notices the weeds, and not just how abundant they were, but how they tended to go up and down from one year to the next. I thought I could rely on data that people had collected. People like my neighbor, Janet Duvall, who had pictures. They'd been around for decades here. They had photographs. They had written records of the plants, of the weeds. And so I thought I could build my model from that. And then one day I was talking to Eric Sager, and he mentioned in our conversation the idea of using satellite data. And I don't know if Eric realizes this, but it really planted a seed with me. And I did a bunch of reading on it and a bunch of research. And that's led to the work I'm doing now. So here's a satellite photo of the Kawartha Lakes area. And to use the satellite photos to measure the abundance of the weeds, I take advantage of two things. One is the satellite photos are actually multispectral. They're recording the image across the light spectrum, red, green, blue, but also infrared. And it turns out that photosynthesizers are like, I guess I think of them as selective foragers. They're pretty good at absorbing and using red, green, blue light. They're not so good at absorbing infrared radiation, that light energy. So they reflect a lot of that back, and we exploit it, as you can see here. The more infrared we see reflected back to the satellite, the more photosynthesis is going on. So I use the amount of that to quantify the amount of vegetation that's growing in the lake. The other piece of information I needed for my model was depth in the lake at every point. So I needed what we call a bathymetric map, but I needed it to be digital. So there's lots of old MNR, MOE, lake survey bathymetric maps available, like this one of Lower Buckhorn. And I turned that into a digital map, so I know at every point in the lake what the relative depth is, what the depth was when that lake survey was taken. So I'm just starting to model now, and I put together the abundance of vegetation as measured by the satellite back in 2018, along with the depth, and that helped me predict to a pretty good precision what the abundance of vegetation was at every point in 2020. So I've made a little bit of progress, as I've shown you, with the satellite work, but also needed to sort of ground truth the satellite information. So I thought I needed to get a closer look at the lake at the same time the satellite was getting its image. So I bought this drone, and the drone takes the same kind of multispectral images that the satellite does. And so I'll be able to relate what the drone is seeing to what the satellite's seeing. So I pre-plan a mission on this iPad app, and then I fly the drone, and it takes all the photos that I then look at in the same way that I look at the satellite photo. Okay, getting ready to run the mission. Here we go. So thanks everybody for listening to my talk. I put my email address up there. Please contact me if you have any questions, and especially if you've got some historical information, images, written records of how the weeds were, other environmental observations. Love to get them. Thank you so much again for listening.
Thank you very much for that presentation, Bob, and for doing our first dockside chat. I think that everybody who lives or has a cottage in the area has a great interest in your research and you know, sort of where we've been, where we are, and where things might be going. So thank you very much for that. Folks, this is a great opportunity to ask Bob about his research. I see there's a question from Greg Finley. Greg, would you like to unmute yourself and direct your question specifically to Bob?
Hi, Bob. Yeah, I would like to know if starry stonewort would have a unique spectral signature that you could see from a drone, for it would make our tracking of it somewhat easier.
Great question, Greg. And it sort of gets to that, what I was saying at the end there about using the drone to ground truth the satellite. And you can see my backdrop here. You get a sort of I think a good idea of how coarse the drone is. What you see just off my, I guess it's my right ear, that's my dock that the drone took off from when I did that little mission. And the big squares that you see are 30 by 30 pixels from the satellite image. So you can see from that, the satellite image obviously isn't picking out starry stonewort. The drone image, I'll wait till next year to give you my story. I've got a lot of work to do on that this year because we also have to ground truth the drone images. Let me give you a picture if I can share my screen for a second. Anna, could you just let me in for 1 sec? Anyway, the drone images themselves, and I'll show you one of them. So, you know, you get a somewhat of an idea. This is pre-macrophyte growth. At the detail is down to centimeters, but to sort of ground truth that to get at what's actually there in a given spot, I'm using another bit of equipment, which I have here. I'll put it in front of me so you can see it. Just a little GoPro camera that my student and I will actually get in the water. That's the way you get in the water these days. And so look at who's there in a particular area that the drone is looking at. So kind of go backwards. There are people that use those drone images now, colleagues of mine, some at Queen's and a couple of other places, to identify harmful algal blooms. You know, we've all heard about those because, again, the spectral signature, multispectral signature from that's pretty distinctive. So a long answer to a short question, Greg. Janet's asking me if the drone came back. So Janet, this is evidence that the drone came back. And I just want to, just on that note, if I can, I'll just share my whole desktop. I don't think I have anything incriminating here. And I'll show you a couple of shots just to kind of give you a picture. So that's one shot that the drone took. But as I mentioned in the video, when you do that detailed work, you're actually putting together hundreds of drone photos. And this kind of gives you an idea, the sort of post-processing, they call it. This is just a small chunk of the shoreline I did there. You can see my dock at the bottom here. I think you can see my mouse flailing around there. But these, I think they're blue boxes up here, is just showing you all the places where the drone took a photo, and then those photos get knit together into the detailed photo that you see. This is another way to imagine how, you know, why I need those drone photos. This is Lower Buckhorn as just a set of thousands of those 30 by 30 meter pixels from the satellite image. And you can see the little black circle up here is that little spot where I took the that test drone image. There's the close-up of it, which is just my backdrop here. So yes, the drone came back, Janet.
Excellent. Thank you for the question, and thank you, Bob, for your response. Does anyone have any other further questions of Bob on his work?
Jeff here. I was just wondering, Bob, if you had an equal water depth and you had different kinds of weeds growing, are the ones that are closer to the surface, like the milfoil, going to stand out? And I just wondered about the water depth and the actual depth of the top of the weeds that's reflecting the infrared light.
Yeah, and thanks for that, Jeff. There is a lot of devil in the detail here. So one thing is those drone images, which I have a student assistant collecting images from, you know, as far back as we can get good multispectral. But the satellite and the drone images, what they're actually seeing in terms of reflectance is being affected by, you know, how the weeds, how the macrophytes sort of layer in that water column, but also what algae are blooming or not during the, you know, other things that are in the water column affecting that reflectance of IR. I also have to, you know, when the satellite happens to come by, as some of you will know, Landsat images are done amazingly, done on every point on Earth every few weeks. And that's what enables me, and I can get that data for nothing, for free from USGS. But conditions differ each time that satellite passes over and gets my image of Lower Buckhorn Lake. So I have to kind of correct for what conditions were on that day. So there's a lot of stuff, including what you mentioned, Jeff. You know, what's the layering that's present. And that's why it was critical for me, if I want to build a credible model that looks to the past, explains the past and helps us explain the future. Got to ground truth it with that drone and then the GoPro right in the water. Yeah, I know we got to get out of here, Doug. Yeah, Andrea's at Ontario Tech with me, and yeah, I'm on a lot of our students' committees, and so yeah, we work together a lot because we're the two sort of aquatic ecologists, field ecologists there. So I got huge respect for her. Wonderful scientist.
Wonderful. Bob, thank you very much. Thank you to everyone who's asked a question. I'm going to ask now if we could go back to Anna and have the next slide, please.
Here we are. That's all good, Tom?
Yeah, that's perfect. Okay, so we're just about at the end now. I'd like to, before signing off, want to thank everybody for taking the time out of this beautiful morning to join us. You can stay involved. There's our new email address. This will make it much easier for us to communicate with you and for you to communicate with us. We've also got the Facebook and the website there. And if you could go to the next slide, please, Anna. Here is, as Ed mentioned earlier, this is our 20th anniversary water quality report. It is just being finished now. We were waiting for the LPP data and finally decided that we would proceed this year and not include the LPP data in the hard copy report. So the soft copy report will be available and will be posted to the website in the coming days. So by this time next week, it'll probably be on the website. We'll be going to hard copy printing shortly thereafter, and we'll make those copies available and we'll make you aware of them. And I'll be mailing out copies to all of our supporters. We'll be sure that the people who advertised with us this year get hard copies if they're open during the pandemic. And I'd like to take this opportunity as well to ask folks that if you would please consider making a donation to Kawartha Lake Stewards. Everything we do is as a result of your volunteer efforts and your financial support. So on behalf of the Kawartha Lake Stewards Association, I would like to sincerely thank you for all of your support in every which way, and hope that you have a really safe summer up at the lake. We can leave, I think we can leave this link open for a while yet. If you could, Kim, if there's anybody who's on the call today and sees someone else's name that they want to connect with, we'll just sort of open it up. Feel free to unmute yourselves and to put yourselves back on video if you'd like to have a conversation. But I'll sign off now, and thank you very much, everybody.
Thank you, Tom.
It's Jeff here. I was just going to mention that, well, we don't have the LPP data in the annual report that's being processed right now. As soon as it's available to KLSA, we will be making it available through our website and a link on our Facebook page.
Good stuff. Okay, that's the end of the formal presentation. If anybody else wants to just communicate, feel free. We'll leave the link on for a couple minutes if anybody wishes to speak to someone else on the call. Thanks, everybody. Have a great day. Bye-bye.